So yes, so I hope you have gone through the week one lectures in the NPTEL website, and so there, so so the faculty who is teaching the course is Professor Mainak Das from Department of Biological Sciences and Bioengineering at IIT Kanpur. And so on, we will be basically talking about whatever he has covered in the week one lectures, the 5 lectures in the week one. And if there is any other questions, queries, or any doubts, we'll be covering that as well. So let us begin. So what is cell culture basically? So cell culture or tissue culture, it is a process or it's a technique by which cells are artificially grown in laboratory conditions under controlled environment. So basically in our body, we have different kinds of cells. We have the neuronal cells, we have heart cells, we have cardiac cells, we have skin cells, blood cells, etc. But all are present in our favorable conditions inside the body at 37 degrees Celsius, and they are all interacting with each other, cross-talking with each other, and they have different functions which are specialized for their cell types, and they coexist very happily. But what we mean by cell culture or tissue culture is we take out certain types of cells from our body, like for example, we take out certain neuronal cells and or certain cardiac cells, and we isolate it in such a way that there are no contaminating cells, like we do not have cardiac cells and the neuronal cells growing together. If that is so, then we call it as co-culture. But generally, cell culture or maintaining a cell line is specifically for a particular cell type. So it's a process by which these cells are, taken out, isolated from your body, and then isolated from the body of animals or plants, and grown in the laboratory conditions under the controlled environment. Anybody has any question?
No, ma'am.
Okay. So yeah. And so this technique was first introduced around the 20th century, and so people used to isolate the explants from plant or different tissues, and they used to study what were the characteristics of such tissues. It was basically a tissue culture, not correctly cell culture, because cell culture particularly means when we are growing cells isolated, like we are growing isolated cells and not a clump or a tissue which has to be grown. So cells were, this the term cell culture, the tissue culture was first introduced around this 20th century, which is long time ago, and they used to study tissue growth and vaccine development, etc. Like because cells, they produce antibodies, so these antibodies can be used against different kinds of diseases, and so people used to study all those kind of vaccine development. Even now, people use cells for culturing different chemicals, different compounds which are of commercial use as well as for the scientific benefit. So it's a widely used technique worldwide, and we use it to conduct experiments because cells are expected to be a homogeneous clonal population. What do we mean by homogeneous clonal population? So it's not very different from what we specifically isolate from the body of any animal or plant, because when we are isolating a particular cell type, so we expect it to behave similar to what its parent cell type was. If it is not behaving similar to its parent cell type, then we do not consider it as a proper cell line. So this homogeneous clonal population of parent cell makes it very important, makes the cells a very important criteria in order to conduct experiments. Anybody has any doubt till now? Okay. So yeah. So homogeneous clonal population of the parent, like the cell lines should ideally. Yes. Rohit, do you have any question? Okay. So as I was saying that the homogeneous clonal population that we are expecting the cells to have, and like it has to be similar to the parent cells. So this is a very important criteria in order to carry out our experiments and in order to design our experiments. Anybody who has switched on the mic, kindly please switch it off. If you have any questions, please ask me or else switch off the mic. Okay. So so this is a very important criteria for carrying out the experiments, because if you want to look at a phenomena associated to your neuronal cells, then you need to have an isolated neuronal culture. If you are interested in kidney cells, you need to have an isolated kidney cell line. So similarly, so this homogeneous clonal population of the parent cells makes it a very important criteria for selecting a cell line for your study. Also, why we use cell culture generally is because it is very feasible to carry out any kind of alteration in the genetic expression in the cell lines. For example, if we want to overexpress a particular protein, or if we want to see what happens in the absence of a protein by knocking it out or knocking it down. I hope you know what is the difference between knock down and knock out. So if not, I will just explain. So overexpressing a protein means you are expressing the protein higher than what it is normally present in your cells. Knocking down means reducing the expression of the proteins in your cells. Knocking out means totally removing the protein from your genetic system, like from the DNA, the entire protein coding sequence is removed, so that your protein is not even translated. There is no presence, no trace of protein at all. So we can alter the genetic expression of cell lines using methods like transfection. So transfection means when you are giving some external DNA to the cells from outside. So that can be in terms of a plasmid which has your protein of interest, or it can be in terms of a CRISPR plasmid which has a gene which has a target sequence target sequence to your protein of interest, and can go and cut in the genetic in the DNA of your cell, and will result in knockout of the protein. So we can alter such kind of genetic expressions in the cell lines using such methods, and therefore, we can see what is the effect of knocking down, knocking out, or overexpressing a particular protein in a particular cell line. So this is very reproducible as well, because cell lines, as I said, is a homogeneous clonal population, and can be used for different kind of studies, checking the expression of the proteins, checking whether the drugs are working, checking whether your antibody is working, etc. So the basics of cell culture, lab etiquettes, and handling of cell culture need to be known to perform better experiments in the future for everybody, for those who are learning cell culture for the first time, those who are going to go for a PhD or postdoc, or going to be associated with cell culture, needs to know certain basics. And this course is all about what are the basic fundamentals of cell culture, apart from being very regularly used techniques in most of the labs. So these are some of the methods of how we basically isolate cells. So this is a human body. For example, they are taking a human body, And cells can be isolated not only from human body but also from plants, but also from prokaryotic systems. There is also bacterial culture that people do, so always there is a confusion between what is transfection and what is transformation. So transfection is when we are giving an external DNA to cells, mammalian cells, whereas transformation is when we give an external DNA to bacterial cells. So there is a so do not get confusion by do not get confused by two terms, which is transformation and transfection. So yes, so transformation is used in case of bacterial cell cultures. Now we have the workflow, how the cells are basically collected. So this is a human body, and we can collect any tissue from the human body, whether a cardiac tissue, skin tissue, liver tissue, lung tissue, etc. So the tissue is collected from the body by scraping off your organ from where you want to collect the tissue. And after collecting the tissue, so tissue has a lot of cells, right? See, you can see there is a lot of cells which are adhered together. I mean, they are clumped in a particular part, and they do not have isolation. Like, this is cemented together. So all these cells, they basically function as a group, so as a population, and they perform a certain kind of function. But in case of cell cultures, we want these cells to behave, to be present in an isolated form, and to have individual characteristics, which is not like functioning together as a group, but it should be able to perform those functions individually as well. So what after collecting the tissue from the human body? So after collecting the tissue from human body, because the cells are all integrated with each other, and they are closely associated with each other, the cells need to be disintegrated, and the connection between each of the cells need to be broken or ruptured. So there are different methods for breaking this connection. There is mechanical disintegration, enzymatic dissociation. There are certain chemicals which can which can break the connections between these cells. So once the connection is broken, then we can see that instead of a group, we get isolated cells. So when we get isolated cells, these cells can be grown into two different ways. One is first culturing. So when we are first isolating the tissue from human body, and then culturing it in a conical flask having certain media and under external conditions, in vitro conditions. So this is known as primary cells, because you are because this is the first set of cells that you have isolated directly from the source. So this is the first set of cells that you have directly isolated, and therefore this is the primary cells. Once these primary cells are growing or is under certain kind of induction, so these primary cells can undergo certain genetic manipulations by either inducing it or by natural spontaneous way, like cancer cells undergo certain genetic transformations. So if such primary cells either naturally or by methods of inducing induction undergo any kind of genetic transformation or genetic manipulation, then what will happen is these primary cell lines will grow continuously, and infinite amount of time it can be passaged, or it can be divided or subcultured. So such kind of cell lines is known as continuous cell line. Here also we are seeing the term transformation is used. So this transformation is not similar to what bacterial transformation means. So bacterial transformation, as I said, it is when we are giving DNA outside, or like from the outside to the bacterial cell. But here what transformation means is changing of the phenotype, like changing of certain kind of expression of certain genes especially the cell cycle regulator genes, which regulate the cell cycle and help the cell to stop after a particular period of time. So in such cases, when those cell cycle regulator genes are affected or mutated, then what will happen is the cell cycle will continue for innumerous number of time, and you can culture those cells, and you can for n number of times you can grow those cells and carry out your experiments. But if such transformation, so that is the meaning of transformation over here, like changing of the genetic constitution of the particular cell in such a way that modulating it in such a way that it can grow continuously, not giving any DNA or external DNA from to the cells. But generally what happens is these primary cell lines, okay, after certain passages or after certain subculturing, so we call, whenever we are taking out, whenever we are subculturing the cells, like from one dish, if you're taking it to another dish, that is called subculturing basically. And that subculturing also in cell culture terms, we call it as passaging. So whenever we do cell culture, we passage the cells, we call it as passaging of the cells, and we always mention that in what passage number we are using the cells. Passage number plays a very important role, because in later passages, like if you are passaging the cell for a very long period of time, in later passages, the cells tend to get stressed, the cells tend to get, their expression of certain proteins will be altered, so you will not be getting the desired results. So it is always recommended that we need to see, or we need to maintain the passage number from what passage number of the cells in which we are culturing. So yeah, so primary cells, if they are not getting transformed, then ideally they will be dying after certain passages, because these cells, they do not have any kind of genetic alteration, and so they will not be, they will not have any kind of so they will behave as normal cells, like how contact inhibition. So basically all normal cells, they die after a certain time. They have a telomere which keeps on decreasing, and they also have they also get exhausted of all their protein, all their essential components which is needed for sustaining their growth. So after a particular passage number, the primary cell line tend to die, and then we call it as finite cell line. On the other hand, if the primary cells, they continue to grow and does not get differentiated, then we call it as a continuous cell line. So then we will move on to what is the difference between primary cells and a cell line. So primary cells are directly obtained from multicellular organisms, and they are grown in appropriate media under suitable conditions. So how I said that is, whenever you are collecting your tissue directly from the source organism, so this tissue, after mechanical disintegration or enzymatic dissociation, the first set of cells that you get, and which is cultured in an appropriate vessel in an appropriate media, that is known as primary cells. And what do you mean by this? These are certain terms that we use, and I felt it should be known to you guys. So what do you mean by confluency? So confluency is a term which we use to tell how much cells are present in our entire surface of the culture vessel where we are growing it. So for example, if we have so this is called as 60 mm. So there can be different kind of dishes. So we have 60 mm dish. Okay, 60 mm dish means the diameter is 60 millimeter. Or we can have a 35 mm dish, which is a little smaller in size. Or we can have a T25 flask. T25 flask looks like, you must have seen this kind of flasks. So it looks like this. So so T25 flask means 25 millimeter surface area, like that. So depending upon the surface area, so for example, if I say, depending upon the surface area, how much amount of cells are present. So if cells are present like entire 60 mm, sorry. So if the entire 60 mm is covered with cells, then we call it as 100% confluent 60 mm. Okay, 100% confluency. So it is not like, so if your 60 mm is 100% confluent, that means your total number of cells which is growing in your 60 mm dish is totally covering the entire surface area. There is no gap between the cells, and that is not good. That is not an ideal condition. Whenever we are culturing cells, it is not recommended that the cells reach 100% confluency, because if the cells reach 100% confluency, there can be different kinds of side effects. One is the cell becomes like the media becomes very toxic, because all the cells will secrete out certain toxic components. They're respiratory, they're like different kinds of toxic components they will be secreting out. Also, when the cells are in very close contact with each other, they will show contact inhibition, and then the cells will not be able to grow much, and they will start dying. There will be a lot of stress. So it is not at all recommended that 100% confluency, we should not allow the cells to reach 100% confluency. Before that only we should subculture it. So the term confluency is used for that how much cells are totally covering the substratum or growth area in the culture vessel. So if it is covering 80% of the total surface, then we say it at 80% confluent. If it is covering 40%, then it is 40%. So once the cells attain confluency, certain confluency, then we around 60 to 80%, like around 60 to 80% confluency is ideal for subculturing. We should not allow it to go till 100%. If we have any experiments, so for experiment models, I personally think that we need to subculture it around 60 to 80%. And if we are doing any kind of microscopic imaging kind of experiments, we have to have lesser confluency, so that we can get isolated cells, and we can select those cells and do our image analysis or image processing. If we are having different, like, so basically if you are doing Western blot, or if you want to check the expression of a certain protein, then around 80% to 90% confluency is recommended. Like, you can have a lot of cells, so that it's not essential that the cells have to be isolated. It is essential that you need to have a lot of protein. So depending upon what your experiment demands, the confluency can alter, but it is never always recommended for 100% confluency. Like, the dish should not reach 100% confluency. Before that, you need to subculture it. So yes, that is what is confluency. Next, what is cell line? So as in the earlier slide, I was saying that this tissue collection, after collecting tissue from any kind of organ from the human or any source organism, when we are mechanically disintegrating it, and we are getting the cells, these cells, if it can passage over like 5 passages, say, and can be unlimited time, and can grow unlimited amount of time, unlimited. Then we call it as a cell line. So cell line is when the primary cells, they are subcultured or passaged. Okay, so subcultured or passaged, for the first time, it becomes a cell line. So whenever it is subcultured, whenever first subculturing you are doing, it becomes a cell line. But there are certain primary cell lines, for example, neuronal cells or cardiomyocytes, they will not last even one passage. So whenever people isolate neuronal cells from mice or from any kind of like organism, zebrafish or something, they will culture it in a dish and they will do the experiment and discard it, because those neuronal cells get differentiated and they do not divide after that. We will talk about what is differentiation, what is de-differentiation in the later slides. But for now, you keep in mind that once the cells, once the primary, like generally the primary cells, they cannot grow for the lifetime, because they have they get differentiated and they have certain specialized functions. So this is also known as Hayflick limit. So Hayflick was the scientist who had, found out this concept that after a certain period of time, the cells stop growing, and that is known as the Hayflick limit. So yes, so primary cell lines, if they are growing, if you can culture it over the period of time for a longer, for unlimited amount of passages, then it becomes a cell line or a secondary cell line. And when this culturing process continues, so whenever the primary cells are transformed and it can become a continuous cell line, then what happens is, so yes, cell line can be two types. So finite cell line and continuous cell line. Okay, so finite cell line is also like a cell line only, but it will not grow after a certain time. But continuous cell line will grow for unlimited amount of time. Uh, so as this, so for this particular point is for secondary cell line or continuous cell line. So whenever the culturing process continues, the cells with highest growth capacity, they will thrive. So for example, so we are isolating certain tissues, and from that tissues we get isolated cells. So these are isolated cell 1, cell 2, cell 3, like that. So out of this, all the cells might not grow, but one of the cell will become transformed, because it will have certain kind of genetic alterations, and that will result in the growth of continuous cell line. So the cells with highest growth capacity thrives, and as a result of which genetic transformation will take place, and it will lead to the development of a secondary cell line. So what is a primary cell line? Primary cell line is when we are isolating cells directly from multicellular organisms. What is a cell line? Basically, whenever those primary cells, you are subculturing it for the first time, then it becomes a cell line. It can be finite cell line, it can be continuous cell line. In continuous cell line, those cells which have the highest growth capacity, and they will thrive, and they will continue to grow until and unless certain they will continue to grow for n number of times, and that results in the development of secondary cell line. I hope it is clear. So next we move on to what are the different types of primary cell line. Like primary cell line can be divided into different types based on different criteria, different characteristics. So on the basis of cell differentiation, the primary cell primary cell lines can be classified into two different types, like differentiated cell lines or stem cells. So primary cells, by virtue of the characteristic of cell differentiation, can be divided into two categories. So differentiated cells are those which have lost the capacity to further that has lost the capacity to further differentiate or divide. So these cells are mainly neuronal cells. The neuronal cells or cardiomyocytes. I know this handwriting is not good. I hope. Okay, so differentiated cells are those which have lost the capacity to differentiate, and they will not divide after a certain point of time. So these are neuronal cells or cardiomyocytes. So so in case of neuronal cells, one of the features of how you can identify differentiated from non-differentiated cells is, in case of neuronal cells, we will see long branches, axon-like fibers, or long differentiated branches. There are certain neuronal cell lines which are not differentiated, like for example neuroblastoma cell lines, or for example glioblastoma cell lines. These are undifferentiated neuronal cell lines, but there are certain cell lines which get differentiated, like primary cells, which is directly isolated from your brain, and direct, like they do not grow after a certain passage. And there is undifferent, so yeah, so one is differentiated cell culture, and another is stem cell culture. So stem cell cultures are those culture, like if you are culturing any kind of hematopoietic stem cells from, if you are isolating hematopoietic stem cells from the mouse and then culturing it over a period of time, then it will continue to grow up for unlimited amount of time. So that is stem cells. Also there are certain cancerous cells. Okay, so whenever we are isolating any cancerous tissue, that has naturally by default lost its capacity to stop growing, and it will continue to grow. So those cells basically are also considered as continuous cell line, and there are different cancer cell lines which people work with all the time. One of the most commonly used cell line is the HeLa cell line, which was isolated very, very long back, and I'm sure you all know what is the story of HeLa cell line. And yeah, so those all cell lines, these are cancerous cell lines and all, they grow unlimited amount of time, and therefore this can be used for different kind of experiments over a long period of time. And people have been using this for quite a while now. And also there is another category by which primary cell lines can be divided. That is their property of like adhering to the surface. So some cells, they prefer anchorage. So they will adhere to the substratum. So whenever we are giving any kind of media and we have a particular dish, for example here in the 60 mm dish, so the cells will go and attach to the bottom of your dish, and we can see that the cells do not move. Okay, so when we have this 60 mm dish with us, we will see the cells are adhered to the surface, and if we move the 60 mm in the microscope, you will not see the cells moving. So so the cells that will be moving are actually dead cells in case of adherent cells. So that is what primary cells which are adherent should look like. Whereas cells which are having cells which are in suspension, for example the red blood cells, the macrophages, monocytes, etc., they will keep on floating and they will not be adhered in the media. And this is a little tricky to culture these cells, because you cannot, so when we do cell culture, and what do we mean by passaging is, we ideally remove all the cells from this particular dish, and then we centrifuge it and we get a pellet. That pellet we resuspend and put it into a next dish. So when we are having culturing cells in a suspension, it is very difficult, because you will not be able to get a proper pellet. You will get some pellet, but you will not be able to get proper pellet. And this kind of cell culture is little tricky. Mostly we in our lab we work with adherent cell lines, and these cell lines include most of the cells, like the human kidney cells 293T, HeLa cells, the glioblastoma cells, there is U87Mg. So these are certain names of the cell lines which is like which we work with. And yes, so depending upon their characteristic of adhesion, they can be different, and they can they can be either adherent cells, or they can be in suspension. Also, depending upon their cell type or morphological structure, there can be different kinds of primary cells. This is epithelial cell line, which looks like this, and these are endothelial cells. So basically, when you start cell culturing, then there are certain morphological features of different cell lines by which we can identify them. But cells of similar lineage or cells which are like, for example, all the neuronal cells might look similar, but because they are all neuronal cells, all the cardiac cells might look, there are different regions of the cardiac tissue, there is different regions of the neuronal tissue, but still they might not be very morphologically distinct. But if you're taking cells from the skin if you're taking cells from the skin, from your skin, and if you're taking cells from your kidney, that might be very different, and you might be able to see morphological changes. So these morphological changes are very evident if the cells have been taken from different parts of your body or totally different organs. Actually there we can actually differentiate between the cell type. Otherwise, morphologically, for very closely associated cells, it is very difficult to identify it. So these are all the different kinds of cell type that cell morphological characteristic. For example, epithelial cell will look like polygonal shape, and it will appear flattened. It will attach to the substrate and form a continuous thin layer. Endothelial cells will be rounded, have a round outline, will not form any kind of sheet or attach to the substrates. And fibroblast like will have like angular shape. This will have fibroblast like features, and it will form an open network of cells that will attach to the substrate. Lymphoblast will be small spherical shape structures. These are lymphoblast, basically is a suspension cells. You will not be seeing it adhering to the surface. If you move the 60 mm dish, it will move along with it because it is not adhering. So yeah, so you will see a small rounded kind of structures. Also neuronal cells look very beautiful under the microscope, because you can see the extensions, you can see their branching, you can see their differentiated structure. But neuronal cells, they are really difficult to work with because of different kind of reasons. Like they are very difficult to get transfected, they are very difficult to maintain, because they get easily differentiated after a certain point of time. So yeah. So there are certain kind of advantages and disadvantages as associated with primary cells and secondary cell culture. So again, I'll just like to revisit. So what is primary cells? Primary cells is when you are directly collecting the tissue from your organism and culturing it. So what does this mean? That this cells directly is associated with the in vivo system. So the difference that these cells will have as compared to the difference with respect to the parent cell that these cells will have will be very, very less as compared to those cells which have been passaged for a very, very long time. So the primary cell cultures for like studies for these are very very reliable source of information. People tend to study primary cells and tend to get their experiment done with primary cells, because they are very similar to their parent cells, and their karyotyping, their genetic composition and everything is very similar to their parent cell. But over the period of time, because of different factors, different handling or different media composition or different kind of external factors, those continuous cell line which is growing, like secondary cell line which is growing, will undergo different changes. And that will lead to having several differences in their genetic composition, and that will result in ambiguity of your results. So there is always an advantage to one part and disadvantage to another part. For example, for like in case of primary cells, if you are doing an experiment, so you need to isolate the cells regularly from the organism, do your experiment, throw the cells. But on the other hand, on the advantageous part is your experiment is much more like comparable to the real life scenario, but the disadvantage is you have to continuously isolate the cells and throw it, and there is contamination issue as well. But in case of continuous cell line, you don't need to continuously dissect the animals and isolate the tissue. These cell lines will grow on for unlimited amount of time. You can passage it and carry on with your experiments. But disadvantage is that as you are passaging it for a very long time, it might get the genetic makeup might get changed, and so you might not be able to have like proper explanation to your results. So those are the advantages and disadvantages of primary and secondary culture. And what kind of experiment you are planning to do, what kind of what kind of interpretation you want to have from the data depends upon what kind of cells you are using, and it all depends upon what kind of problem you are working with. So next, let us talk about cell cycle and differentiation of the cells. So we are all, so I'm continuously mentioning that cells get differentiated, cells do not grow. So what do we mean by differentiation of cells? So we have to go to the basic of cell cycle. So whatever cells we are getting now is getting divided by mitosis. So mitosis means that when one cell gets divided into two cells, and those two cells are identical to the parent cell because there is no mixing matching of the genetic material. So these are identical clones of the parent, and therefore these cells continue to, like, whenever they are dividing, whenever they are undergoing mitosis, the clonal population should have identical features to their parents. But why do they get changed? Because there might be mutation, there might be different external factors for those reasons. But ideally, anybody has any question? Suryakant, you have any question? Your mic is on.
No, ma'am, continue.
Okay. Can you please switch off your mic? Thank you. Okay, so yeah, so I was talking about what is cell differentiation, and why do we need to, why do we, the cells that is growing, that is clonal replicates, this is identical clones of their parent. So cell cycle, if we are well versed with cell cycle, so what happens is there is a phase known as interphase. So in this phase, the cell will gather all the material, all the nutrients, all the genetic material, all the accessory part for undergoing the particular cell cycle, undergoing the mitosis. There is the first phase of cell cycle is interphase. Then it enters in interphase we have G1, S, and G2 phase. So in this G1, S, and G2 phase, all the cellular. So although it looks like although it looks like the cell is not dividing, cell is not performing anything, but actually a lot of undergoing molecular mechanisms are being followed inside the cell, and so one of this is in G1 phase, all the cellular contents, including the excluding the chromosomes, are duplicated. Everything inside the cells, the nutrients, the proteins, everything, etc., is duplicated except the chromosomes. In S phase or synthetic phase, the 46 chromosomes which is present in the human mammalian cells, that gets duplicated. So we get two pairs of 46 chromosomes, and so it prepares. Basically, the cell is preparing for it to get divided. Then in the G2 phase, cell will double check whether everything has been followed, whether the chromosomes have been duplicated, where the nutrients have been already doubled, whether all the checkpoints have been passed. So there are certain checkpoints which regulate, the cell to go from particular one phase to another phase. There is G1S checkpoint, G2M checkpoint. There are different checkpoints. So all those checkpoints have certain criteria or have certain list of things which the cell checks before it proceeds to the next phase. And if it is not followed, then the cell cycle gets stopped. So all these are natural process in our body. Skin cells continuously they undergo this kind of mitotic division. So G1S and then G2. Finally, the cell enters the mitotic phase, where again we have four kinds of where we again have different stages: prophase, metaphase, anaphase, and telophase. And the cell gets finally divided into two different identical clones. So one cell gets divided into two different cells. So here we can see the schematic representation. So here we have the first, so this is one cell which has one pair of 46 chromosomes, everything else is duplicated. Then in S phase, we get two pairs of 46 chromosomes, and then we get in G2 phase, we will see whether all the checkpoints have been met clearly, and then the cell will undergo mitosis. So once it undergoes mitosis, then we will get two cells which are identical to each other. But there is also one more step in the mitotic cycle where the cell cycle gets arrested, and this is known as the G0 phase. So the cells which do not divide or which get differentiated, which we say is differentiated because they have attained a certain cellular function, is because they enter the G0 phase. The cell cycle is completely arrested over here because there is no other like they have attained certain specialized function, and they cannot revert back unless it is like another stimulus is there. They will not be able to revert back to the mitotic cycle, and they will stay there as the dormant stage and will ultimately die. So this is the G0 phase where the cell gets differentiated and the cell attains specialized function. So this particular stage is known as differentiation of the cells. Otherwise, all the other cells will get divided and will undergo mitosis, and the cell cycle continues. And those cells which undergo this mitosis continuously, those are known as the continuous cells, and they will form the continuous cell line. And those they get arrested over here, they will form the finite cell line. Okay. So there are certain terminologies which I thought that maybe I'll discuss. So what do you mean by differentiation? So differentiation is when cells have developed a special function. So neuronal cells, they get differentiated because they have a special function for conducting the nerve impulses throughout the body, and for brain impulses and everything. So they will not be dividing after that. Once your neurons are formed during your childhood, they don't get divided, and so they get differentiated. So those cells get differentiated. This is what we mean by differentiation. What do we mean by de-differentiation? So de-differentiation means when those differentiated cells loses its specialized function. So there might be some cases where your cells, they tend to lose its specialized function because of some kind of stimulus, some mutation, or some requirement of the body. And if it loses those specialized function and can revert back to its original capacity to differentiate, like original capacity to divide and differentiate, then it is known as de-differentiation. So again, I will just repeat. just to not get confused. So differentiated cells, if it loses its specialized function and regains its ability to differentiate or regains its ability to divide, then it is de-differentiation. And what do we mean by de-adaptation is when cells lose partly their differentiation ability, like not fully, they can still differentiate, but not fully because we are subculturing it, right? So if we are subculturing a particular cell line for a long period of time, then over the period of time, at one point, the cell will become different, like the cell will behave like it is differentiating, although it will still have some property of dividing. So then it is known as de-adaptation. So it is getting adapted to that media, and it is partially losing their differentiation ability, but it is not fully losing their differentiation ability. So next we'll talk about potency. So potency means what is the ability of the cells to generate other kind of cells. That is how potent a particular cell or a tissue is. So there are different terminologies associated with this. For example, one is totipotent. This is number 1. Second is pluripotent. This is number 2. Here it is written as unipotent, but also there is another term as multipotent. This is L, this is T. Multipotent. Okay. So what do we mean by totipotent, pluripotent, multipotent, and unipotent cells? So this is an oocyte, or this is fertilized by a sperm, and we can get a zygote. So zygote or spores, they are known as totipotent cells. Zygote or spores. So we have spores in plant like algae, fungus. We also have spores in bacteria. So all those kind of structures, they are totipotent cells. So totipotent cells means they have the ability to give rise to all kind of cells, irrespective of whether it is an adult cell, whether it is an embryonic cell, every kind of cell the totipotent cell will give rise to. So zygote or spores or the morula, okay, so these are all kind of totipotent cells and can give rise to all kind of embryonic or adult lineages, cells of all embryonic or adult lineages. So once the zygote divides and attains morula, then this morula can become any kind of cell, because you can see that as the embryo grows, this particular from this morula there is differentiation of the ectoderm, endoderm, and mesoderm. So ectoderm, endoderm, and mesoderm. Sorry, this is ectoderm, mesoderm, and endoderm. So all this kind of distinguishing the different kind of tissues inside the body of the cell, inside the body of the organisms, they kind of take place from this totipotent cells. And once these cells, grow in a particular stage in the embryo, so these are the embryonic stem cells. So once they become the embryonic stem cell, this is the human fetus, and become the embryonic stem cells, those cells, they are able to give rise to all kind of adult cells, but not embryonic tissues. So embryonic tissue, extra embryonic tissue includes placenta. Also, there are embryonic tissue which is the umbilical cord. So those things, they cannot, this kind of cells cannot divide and cannot form those extra embryonic tissues. But this embryonic tissue, but the pluripotent cells can give rise to all other adult lineages. For example, the circulatory system, the nervous system, the immune system, etc. So, what is the difference between totipotent cell and pluripotent cell? So, totipotent cell can give rise to all kind of cells of all adult as well as embryonic lineage, whereas pluripotent cell, they can give rise to cells only of the adult lineage, not of the extra embryonic tissue. So, the example of pluripotent cell is is embryonic stem cells. Embryonic stem cells. And also we call something as iPSCs, that is also pluripotent stem cells. So so that is the difference between pluripotent cells and totipotent cells. Now we will come to multipotent. So basically the zygote is growing to a morula. Morula is a totipotent cell. Then the morula will grow into an embryo, like a small embryo. There all these cells are pluripotent cells. These embryonic stem cells are pluripotent cells. They will not be able to give rise to placenta. Then this grows into multipotent cells, where multipotent cells are basically the hematopoietic stem cells. Hematopoietic stem cells. So potipotent cells are hematopoietic stem cells. So what are hematopoietic stem cells? We get this basically in our bone marrow. Okay. So in bone marrow, we have there is the generation of different kind of red blood cells. There is different lymphocytes, monocytes, etc. All those things are grown in the bone marrow. So this bone marrow helps in growth of different kind of cells of the same lineage. So what do you mean by multipotent cells? Multipotent cells are those which is capable to give rise to different cell types of the same lineage. For example, like only blood cells. This cell, so hematopoietic stem cell can give rise to only blood cells. Okay, circulatory cells will give rise to only the cells of the cardiac tissue. Nervous system will give rise to only nervous tissue. So similar to that. And what do we mean by the fourth criteria is the unipotent cell. So in unipotent cells which only have the capacity to divide into one type, that is the unipotent cells. So in unipotent cells, so cells like the circulatory system, nervous system, immune system, multipotent cells do not come under this. Only hematopoietic stem cells are under multipotent system. Under unipotent, we have circulatory system, nervous system, and immune system, because these are specialized for a particular function, and they cannot revert back to any other cell types. So that is all about potency. Now we will talk about, so we have talked about how we grow the cells, okay, how we have different kind of culturing, different kind of adhesion cells, suspension cells, we have stem cells, we have differentiated cells, stem cells, adherent cells, suspension cells. So how do we continue to grow it over a period of time? It is not possible that you will continue to grow the cells without taking any break, right? So we do cryopreservation of the cells so that we can preserve it under very low temperature, and whenever we have our work, then we can again take out those cells and again start culturing. It is not like that you have to like continuously grow the cells for for innumerous time without taking any break. So for that we use a technique called cryopreservation. And in cryopreservation, the term cryopreservation itself means in cold temperature we have to preserve it. So it is a technique which preserves the organelles and cellular structures by cooling the samples to extremely low temperatures around minus 120 degrees. So like if we go beyond this temperature, then the cells will start dying. And if we go below this temperature also, then also the cells will not be like proper, it will not show proper features, it will not be characteristically similar. So it is very we have to store the cells either in minus 120 degree, not beyond that. And also we have to keep in mind that also we have to keep in mind that we should not during preserving the cells, we should not keep the cells outside for a very long time. So we use DMSO in our in the media in which we preserve the cells. So DMSO is a cryoprotectant, and what it will do is it will cover the layer of the cells, and it will prevent it from crystallization. So if the cells get crystallized, then the cells will basically burst and die. But if there is DMSO, it will not allow that crystallization. It is kind of surfactant, and it will not allow that crystallization, and it will protect it at such low temperature. But DMSO in room temperature is toxic to the cells. So during cryopreservation, when we take the cells and we want to preserve it, then we have that we have the freezing media which has DMSO in it, and we need to quickly put the freezing media in the cells, resuspend the cells, like resuspend the cell pellet, and then put it in a we have one vessel called a Mr. Frosty. So Mr. Frosty looks like this, and I know the name is little funny, but yes, that is the name of it. So this is Mr. Frosty. What Mr. Frosty does is, so when you have DMS, so when you basically you are not supposed to put the vial directly in minus 120 degree, because if you are preserving, like you are doing everything in room temperature in your cell culture hood, and after suddenly adding, if you put it in minus 120 degree without gradual decrease, then the cells will get stressed and the cells are going to die for sure. So we have this Mr. Frosty, where there is some small sockets for placing all your vials. So once we are adding DMSO once we are adding the freezing media which has DMSO in the cell pellet, then we take that Eppendorf tube, and in this Eppendorf tube this is not Eppendorf tube, we have to use cryo vials. So cryo vials can sustain lower temperatures. So cryo vials also have look like this kind of thing. So this cryobias, we have to put all our cells and then put it in Mr. Frosty. So Mr. Frosty, it will reduce the temperature slowly, 1 degree, like slowly by 1 degree. And from room temperature, so once you put this thing in Mr. Frosty, and then this Mr. Frosty needs to be kept in minus 80 degree. So this so the cells from room temperature, slowly, it will reach minus 80. Then you need to take out your cells and then finally put it in liquid nitrogen, which is around minus 120 degree. So the cryopreserved vials are recommended to for long time storage. We need to put it in liquid nitrogen, which is around minus 120 degree temperature. So we have to be really careful whenever we are dealing with liquid nitrogen, because it is so cold. There is chances of getting frostbite, and we have to be really careful whenever we are doing cell culture, because different kinds of chemicals. Many chemicals are very toxic. DMSO is very toxic, to be particular. Then liquid nitrogen is also very, you have to be really careful with it. So cell culture on an average is like not a very difficult technique, but also we need to be very careful while doing it, because there are chances you might not be very focused, and things might get out of control. So that is all for today. Now, before I go to the question, like there are certain assignment questions, not this year's not this course, not this assignment that you will be getting, but some questions that I had earlier. So we will be discussing those questions. Before that, if anybody has any questions or doubt, please let me know now.
Ma'am,
Yes.
can you explain what is partial differentiation?
Partial differentiation means like, for example, so you will have a cell, like some cells which will, so like suppose in your culture dish you have 100 of cells. Out of 100 of cells, say 50 cells are getting differentiated, whereas the other 50 cells are still in the dividing phase. But those 50 cells which are getting differentiated, they are not going to divide further, and as a result of which the like slowly, like over the period of time, over the period of subculturing, all other cells, because those differentiating cells, no, they also secrete some growth factors, they secrete some kind of chemicals which will be, which will help the other non-dividing cells, no, like dividing cells, to differentiate as well. So it is really that is called partial differentiation. Like you have 100 cells, 50 cells are differentiated, 50 cells are still in the dividing phase, but after a period of time, the other 50 cells will also get differentiated, because the differentiated cells will produce some growth factors. So that happens due to subculturing and exhaustion of all the nutrients inside the cell. Telomere gets exhausted, like some things like that.
Thank you, ma'am.
Yeah. So please elaborate transcription. Okay, there are some questions in the chat box. I will go through one by one. Can you please elaborate transfection? So yes, transfection is basically, so I will tell you, let me go to the slide again. So in this transfection, what we do is we have a dish, and in this dish we have cells which is cultured. Okay, so ideally it should be around 40 to 50% around confluency. Okay, so in this 50% around confluency, so whatever protein you want to see their expression, those protein is present in a plasmid. So this is a plasmid. Okay, and you have your protein of interest, POI. I'm just labeling it as POI. So this protein of interest is under a particular promoter, suppose mammalian promoter, okay, which can express in the mammalian system. I'm talking about mammalian cell culture. If you are interested in insect cell culture or plant cell culture, then this protein of interest needs to be under the expression of that particular organism's promoter. So this promoter is, for example, mammalian's promoter, okay, mammalian promoter. I'll just write as P. Anyways, so this mammalian promoter now will, so this plasmid basically, now this is a plasmid. So this plasmid is a expression vector. I'm sure if you don't know what is expression vector means the vector which has the capacity to produce protein, not just the replication part, but also the translation part as well. We have three, in central dogma, we have replication, replication, then we have, what we have? I forgot. Transcription, replication, transcription, and translation. So in case of this plasmid expression vector, it has to follow all the three steps: replication, transcription, and translation. So the plasmid has to get replicated. Also, it has to express the protein. So this plasmid containing our protein of interest is then put inside the cells. So this is the method by which we have to put this particular plasmid containing the protein of interest inside the cells is called transfection. So transfection means here in the cells we are basically this plasmid DNA, it is encapsulated in a micelle. So what do you mean by micelle? Micelle is like small vesicles which are having lipid bilayer. So we in case of cells, the cellular membrane is made up of lipid bilayer, right? So any kind of external proteins or external substance cannot enter this cell membrane barrier. In order to cross that barrier, what the principle of transfection says is we will encapsulate our protein of interest containing plasmid in this micelle. So in this micelle, and this micelle containing plasmid of interest can then now enter the lipid bilayer, because micelle also has the same composition of the lipid bilayer region of your cell. Once it enters the cell, inside the cell, it will start expressing all those proteins and like whatever proteins is there in your plasmid, it will start producing, and then you can check the effect. So this particular process is called as transfection, where you are basically putting the plasmid, like external DNA, inside the cells. Okay. Now I will go to next question. So Revant Kishore is asking, please do elaborate on continuous and finite cell lines. So continuous cell line is basically whichever cell line gets transformed, okay, and there is genetic alteration, and then it can be like primary cells when you're isolating it from the body and then continuously growing it, and it does not die because there is some genetic manipulation, either naturally, for example, cancer cells, it will grow infinite number of times, or if you are inducing any kind of chemicals which will allow those primary cells to grow for infinite number of time, or if by natural, or if by like virtue of its kind of mutation, if there is some changes, then it can grow for infinite time. That is known as continuous cell line. In case of finite cell line, we that genetic manipulation does not take place, and the cells will die after certain passages. So the cells will then, so after that passage, then you have to discard those cells. Simran Patil, ma'am, will this be? Yes, the PPT and the recorded version will be available. We will, so through the NPTEL website only, you can access all the recorded lecture as well as the PPT that I have prepared. What is co-culture? So co-culture is basically when you are having two different cell lines and growing it together. So if you are growing neuronal cell line and cardiac cell line together, then it is called co-culture. I am not very well versed with co-culture, but yes, this is what basically it means. Yes, so you will get the PPT and recordings, so don't worry about that. So differentiation means whenever the cell is entering the G0 phase and the cell cycle is arrested. So, and the cells are undergoing certain specialized function, that is differentiation. De-adaptation means cells are not getting totally differentiated. Okay, there are certain cells which can still divide, and there if those cells are dividing, then we can have like partial dividing cells and partial undifferentiated cells. Those are known as de-adaptation, because it is getting adapted to the cell culture media. Like the natural characteristic of cell is to die, but if cells like get differentiated, and if it is like growing for a long period of time, then it is like de-adaptation, or it is getting adapted to the media. So Matthew John is asking, can we control which cell to become continuous and which to become finite? Yes, to some extent we can control that, because there are certain cell lines like where people are actually inducing the cell to behave as a cancer cell, or to behave as a continuous cell line. Apart from all the cancer cell lines that we already have, there are certain cells which can behave, can grow unlimited amount of time due to some chemical inducers or due to some genetic manipulation that people have done throughout, like people have been doing it. So Revant Kishore is asking, as you said that our culture should be as close as possible to in vivo condition, but in plant tissue culture we have a formation of callus, which doesn't even exist in vivo. So then how are we to interpret the in vivo metabolic pathway? For plant tissue culture, I am not very sure. I will look into it and I will let you know. Okay. Ma'am, can you explain for pluripotent cell? So pluripotent cells are like, for example, hematopoietic stem cells. So which can give rise to certain cell, like which can give rise to different types of cell, but within the same lineage. Same lineage means same type. Like for example, hematopoietic stem cells can give rise to only the blood cells, like the red blood cells, white blood cells, those. Not kind of like all different types of cell. So that is pluripotent cell. Totipotent cell means it can give rise to all different types of cell, whether it is a heart cell, whether it is a kidney cell, lung cell, everything. Totipotent cells are for everything, for the embryonic tissue, non-embryonic tissue, etc. But pluripotent, pluripotent, no, that is multipotent cell. Pluripotent cell means, for example, the embryonic stem cells, which can give rise to the adult lineage cells, like the embryonic stem cells are having this ectoderm, mesoderm, endoderm, but they will not have extra embryonic tissues like placenta or the umbilical cord, those things. So those cannot be given rise by pluripotent cell, but it can be given rise by totipotent cell. Totipotent cell can give rise to all the embryonic tissue, extra-embryonic tissue, as well as adult tissue, but pluripotent cell can give rise to only adult tissues, like cells of the adult lineage, not of the extra-embryonic lineage. That is it. So, ma'am, I have, ma'am, okay, so Samad, you are writing that whether we can, introduce any cell function. So when we are culturing the cells, it is not totipotent cells. when we are culturing the cells in the in our culturing vessel, as I mentioned earlier only, it is a particular cell type. It is not like it can be grown into different kind of cells. We are isolating either kidney cells or heart cells or neuronal cells, not like we will have stem cells which will be like different kinds of, it can grow into different lineages. Those kind of cell culture, people don't like, I am not aware of it. Always people are more into culturing cells of a specific organ, cells of a specific tissue, not like so basically those cells have a particular function. I mean, kidney cells will behave like kidney cells, heart cells will behave like heart cells. It is not totipotent, so there we cannot introduce any kind of function, cell function which will make kidney cells to behave as kind of heart cell. But yes, there can be, because there are different kind of expression. So your heart cells will have different proteins which will express in a different level. In the kidney cells, in physiological condition also, it is not possible that it will behave like a heart cell. So ideally it is always recommended that whatever problem you are working on, if you are working on a neurodegenerative disease, we should work on a neuronal cell line, not on a cardiac cell line. If we are working on a cardiac disease, we should work on a cardiac cell line, not a kidney cell line. So that way it is done. Like in that kidney cell line, what is the effect of your protein? What will happen if it is changed or something? That way, it is not recommended like we can make heart cells into kidney cells and then study the function of kidney in it. Okay. So next is now assignment should be done under. So yes, so it is under minus 130 degree. No, so minus 120 degree is under minus 130 degree, right? So it is correct only. What about Preeti? So yeah, that is your question. What about the cell culture of cells that shows the response to different cells like cardiomyocyte? How does that work? Come under different cell cultures of cells that show the response to different cells. Samiksha, can you please explain? I am not able to understand your question. So yeah, we will be discussing about the questions, not the week 1 assignment questions that you have got. We will be discussing about the other, other kind of similar questions. So that way you have to, you can answer the question and then let me know your answers. So let us. So Karti, this week's questions you have to answer by yourself only. Okay. I will be sharing my screen and share with you some questions. So yeah, Uh, Let us go into some questions. Suryakant, you have any question? Yes, please go ahead. Can you please repeat it? I did not get your question.
I want to know about the future prospect in monoclonal antibody production.
Yeah, definitely. Cells are being used. So there is this hybridoma technology where cancer cells are fused with your B cell, which is producing a particular kind of antibody, and so it can be continuously generated like B cells. If you are fusing it with a cancerous cell line, then the B cell, which is originally supposed to die, will not die and continuously grow and will secrete antibodies, monoclonal antibodies. Like if you want to like against a particular protein, if you want to isolate certain antibodies, so it can grow that way. So it is hybridoma technology that people use for growing monoclonal, like for isolating monoclonal antibodies from cells, from B cells.
Right now, this kind of technologies are and the disease landscape.
So you are not very clear. What again? Can you repeat it? Can you type the question?
Actually, monoclonal antibodies are used for any kind of disease treatment.
It's kind of used in disease. What?
Actually, recently, monoclonal antibodies for treatment of
Not able to understand. Anybody is understand?
actually, I want to know about in any set, we are using for launch and monoclonal antibodies for treatment of any disease.
Still not able to get it. 1 second.
ट्रीटमेंट करने के लिए monoclonal antibodies हम यूज कर रहे हैं recently.
Please type it. I think I'm not able to hear you. Sorry, Suryakant. If you type him, then only I will be able to answer it. Okay, so I'll just go through the questions. Okay, quickly. Let us go through. So you can type in the chat box. I will just see the answers and then tell the answer. So what is the answer to this question? Primary culture becomes what after first subculture? There is no option as secondary culture. There is, out of 4 options, whatever option do you think correct? Just tell me option A, B, C or D.
Ma'am, it is not visible on the screen.
Oh, it is not visible. Okay.
Yes.
Now it is visible.
Cell lines.
Now it is visible? No.
Should be.
Yes, ma'am.
Now?
Is it cell line?
Now is it
Yes,
visible?
ma'am, it is visible. It
Yes,
is
Okay,
ma'am.
visible, ma'am.
thank you.
Yes, ma'am.
Yes. So the answer is cell line. Do not tell it in the mic because everybody will be if everybody starts telling, it will be difficult. So write it in the chat box. Okay. Okay, that is correct. It is cell line. Now, next question. Okay, so Suryakant is writing, recently we are using or not monoclonal antibody for treatment for any disease. There is antibody based therapy for cancer. People are, this is like people are using for like now some quite time, and yes, definitely it is revolutionizing the cancer treatment. So that is why. Now we'll go to next question. So what does 80% confluency means?
Ma'am, question not visible.
Oh, okay. Let me go to. Afreen, can you please switch off your mic? Rakesh, you will be getting the entire PPT in your NPTEL website. Okay, so you don't need to take any screenshot. You will get the recording. You will also get the PPT. Everything will be accessible. So it is fine. Like you don't need to take screenshot. Okay, so the answer is correct. Option D. Very good. So next question. That's correct. Correct. So cell lines that can divide a particular number of times, so that is known as senescence, and these are known as finite cell lines. So correct. Option number A. So this is the next question. Question number 4. Yes, that is also correct. DMSO, it acts like a protective agent, and it will provide protection against crystallizations in the such cold temperature. If there is no protected or cryoprotective agent, then the cells will get crystallized. So yes, Akash, you will get the assignment in the NPTEL website itself. You will get your assignments there only. There only you have to submit. So the correct answer is answer option A. That is right. Next question is, very good. The correct option is on answer C. Very good. Question is about. So Samiksha is asking that whether the cell culture of cells that shows the response with the other cells like cardiomyocytes show electrical and mechanical response with different cells surrounding different cells. So how does that work? So Samiksha, actually, so the inherent property of cardiomyocytes is like it is going to show this kind of electrical impulses, mechanical response, such kind of that is the inherent property of it. If it does not show, then it is problematic. But if it is showing, it should be able to show it in spite of whether it is interacting with other cells or not, because that is the inherent property of those kind of cells. For example, if you see the neuronal cells, if it is not branching, so even in the presence of other cells, or even in the absence of other cells, like for example inside the human body, it is also surrounded by different kind of blood cells or different kind of tissues, but it is going to show the differentiation characteristic there as well. On also when we are culturing it in vitro condition, there also it has to show those characteristics. So it is independent of whether or not it is associated with different kind of cells in the surrounding vicinity to show its inherent property, because this is what makes it a heart cell, or this is what makes it a neuronal cell. That is its like characteristic. You cannot take out the characteristic even if you are growing it outside the body in vitro conditions. So even if you're growing it in vitro condition, it should still maintain those general characteristics. I hope it answered your question. Okay, so next question is, which of the following is a non-adherent cell type? Correct. So everybody has given option number D, which is right. So red blood cells, these are suspension cells. They basically they are total flowing through our body, so it cannot be adherent. So that's the correct answer. What do you mean by anaerobic respiration? This is, I think, very easy question. Correct. So the option number B is correct. So next is this question. So if a terminally differentiated cell type gains stemness, it is called? So stemness means the ability to divide. There is a confusion between redifferentiation and dedifferentiation. Actually, it is option D, differentiation is correct. Number C, because dedifferentiation means, see, if you break the entire word, differentiation means it has differentiated, like the cell has lost its ability to divide. Dedifferentiation means the differentiated cells, they are regaining its ability to divide again. Terminally differentiated cell type example is differentiated cell type example is neuronal cell or cardiomyocytes. Those are terminally differentiated cells. Okay, so dedifferentiated means that differentiated cell line, they have lost its ability to perform the specialized function, because of which it had lost its capacity to divide. So now, because this differentiated cell line have lost its capacity to perform the specialized function, it is reverting back into its original type, where it can again undergo differentiation, or where it can either it can undergo again differentiation, or it can undergo divide, like it can divide again. So basically, dedifferentiated cells means differentiated cell which is reverting back to its original form, where it can either divide or again get differentiated. RBC blood cell is terminally differentiated cell. No, RBCs can grow. RBCs can divide. So one RBC can again divide, right? We have like lifetime of RBC is 120 days. So again, then new cells grow. No, these are not stem cells. Stem cells means those cells which can grow into any type of cells, like hematopoietic stem cells. Those are like any type of cell. But RBCs do not can divide. 1 second, wait. So, yeah, anybody? What about RBC?
RBCs directly die and go to the spleen, right, where they die, and then again the new RBCs are replenished by originating from the hematopoietic stem cells from the bone marrow.
Yes,
But they don't
actually.
divide.
Yes. Yeah, it is not. So RBC is a differentiated cell line only, correct? Because it is like, so yeah, Revant is correct. It does not have a nucleus, and so it will not be dividing. Hematopoietic stem cells will only give rise to red blood cells, and those red blood cells again, like once it dies, again fresh red blood cells get replenished. Correct. So that is my mistake. Sorry.
And ma'am, what is redifferentiation?
What is redifferentiation? So redifferentiation means if it is redifferentiation, I have to check once. These are very confusing terms. Differentiation, redifferentiation, dedifferentiation. So redifferentiation, I think it means like the cells which had differentiated again can differentiate into another type. I think it means that. I'm not sure. Like dedifferentiation means it is regaining its capacity to divide or differentiate again. But redifferentiation means if it is like differentiated in a particular differentiated cell line which is having a specialized function, if it regains its capacity to differentiate into another like another kind of cell line which can have a different function, maybe that.
So here the answer should be redifferentiation, right? Because dedifferentiation only talks about the losing activity of the differentiated cell. It doesn't
Uh,
talk about the division, right? But the redifferentiation says that the differentiated cell can have the ability to divide and have a particular task to do.
No, redifferentiated cells, the differentiated cells, they will not divide. They will become like they can they will again get differentiated into another kind of cell. Like they will not be undergoing cell cycle or mitosis. But dedifferentiation means that the cells can actually undergo mitosis again. Like they will not, they also have a scope where they will not get differentiated and just undergo cell cycle and just like continue to grow.
Okay, ma'am.
At least that is what I have
नहीं
interpreted.
मैं 400 ग्राम का ही लाई हूँ, वहाँ ऑफर नहीं था 1 किलो पे।
Okay. Hmm. So yes, the differentiated RBC is a terminal differentiated cell, correct. RBC die in your cell, the differentiated cell again differentiate is called redifferentiated. Yes, the differentiated cells which will differentiate again is redifferentiated. Is dedifferentiation an irreversible change? It is not an irreversible. Again, it can get differentiated, so it is not. I don't think so. It's a irreversible change. And dedifferentiated cells loses. Can neuron divide? No, neurons can't divide. Neurons are like neurons which are they do not. Once your neuronal cells have been differentiated, they will not be dividing. So yes. So last question for today. This is, and go through the question and let me know. Matthew, is the lecture sir told dedifferentiation is irreversible? Then the concept of redifferentiation confused me. Actually, in the lecture, no, sir had said that dedifferentiation means cells loses its ability to differentiate. That I also have one confusion. In the lecture, he has said that cells loses its ability to differentiate. Dedifferentiation means, but actually dedifferentiation means cell regains the ability to, I mean, divide. That is what I know. So basically just keep in mind that dedifferentiation means cells which had differentiated or lost the capacity to divide have regained the capacity to divide. That is dedifferentiation. And redifferentiation means cells that have differentiated and have lost the capacity to divide regains the capacity to divide, not divide, regains the capacity to again differentiate. That is what I think is like the difference between redifferentiation and dedifferentiation. Okay, don't get, because these are very, I mean, very confusing terms in terms of like even like there are these terms have been introduced in plants and like there are certain tissues which get redifferentiated, dedifferentiated, this that. So yes, and differentiation function of cells become. Yes, Karthik. On differentiation, function of cells become more specialized. That is correct. More specialized. Function of cells become specialized. There is not more or less. That cells are specialized for a particular function or role. That is the meaning of differentiation. Differentiation implies that the specialized properties of the cell are lost irreversibly. Yes. So dedifferentiation means the specialized properties are lost. Yes, this is correct. Samad is correct that dedifferentiation means that the specialized property that the cell had gained, that is lost because now the cell can divide again. How can we induce a differentiated cell to become dedifferentiated? How can we induce a differentiated cell? if there should be some chemical inducers. I think like in natural conditions, maybe some mutations or something have. Like I know in some plants or something, there is this phenomena where differentiation and de-differentiation happens. Like in case of cork, or in case of some like the plant bark or something. I remember that this phenomena, differentiation de-differentiation, happens naturally, but in case of cells or in case of cell line, I am not very sure, so I will not be like giving any wrong information on that. But yes, so I am not very sure on that. Yes. Redifferentiated cells lose their ability to divide but can mature and perform special function. Redifferentiated cells are differentiated cells which are, which can divide again. I will just once check what is redifferentiated. Redifferentiated. Yes. So So redifferentiation happens in this secondary cortex, xylem, phloem on cork that I was saying. No, that it happens there. So redifferentiation means differentiated cells only there, but they are now performing some other kind of, like I remember that in plant cells differentiated to become degenerated. But what do you use in animal cell cultures? Does differentiated cells will become dividing cells? Yes, de-differentiated cells will become dividing cells. Yes, they can become dividing cells. In plant cell cultures, we use auxins. Okay, so Matthew is asking, are IPCs examples of such induction? What induction? Pluripotent stem cells. So they can, yes, they can be induced to become any kind of multipotent stem cells. And pluripotent stem cells can become multipotent stem cells. So they are ideally not getting differentiated. They are again, they can divide again. Pluripotent stem cells can. So pluripotent stem cells either can divide and become again pluripotent stem cells, or they can get differentiated to form multipotent stem cells. And thus differentiated cells will become dividing. In plant cell cultures, we use auxins and cytokinins to make differentiated cells to become de-differentiated. What do we use in animal cell cultures? Revant, I am not sure. What do we use for differentiate? So there are some kind of different. So for example, I know in case of neuronal cells. Okay, so somebody has switched on the mic. Can you please switch off? Okay. So in case of cells, no, in case of cells, for example, in neuronal cells, there is some chemicals like bb-cyclic AMP. Okay, so we have certain. So there is a neuroblastoma cell line called N2A. Okay, this is a mouse neuroblastoma cell line, and this cell line is originally a unlimited or continuous cell line. It will grow continuously. But like if we want it to get differentiated and if we want it to stop dividing because we want to check the expression, or we want to check do our experiments in differentiated cells, then we use a chemical called db-cyclic AMP, which will differentiate this neuronal cell line into the differentiated portion. Like it will not divide after that. So there are chemicals which is specific for certain cell lines like that. So that is your answer. Okay, Revant. So we use certain kind of chemicals, like for example, in our lab we use retinoic acid can be used, or we can use this db-cyclic AMP is used for differentiating neuronal cell lines. I'm not sure what is used for the cardiomyocytes or other kind of differentiated cell lines, but for neuronal cell lines, we generally use retinoic acid. We generally use db-cyclic AMP for differentiating it. Krishna, normal cell differentiate to become specialized cell which are non-dividing. Correct. And what is this? Is induced pluripotent stem cells. Correct. Yes. After how many cell cycles we have to stop the cell line and dispose them? That is so, Madhula. After how many cell cycles exactly we have to stop the cell line and dispose them? It is not like written in books and papers. It is like when you see that the cells are not in the stage to continue to grow, then you have to discard it, because there are cell lines which have been growing for several years now. So it is definitely not like it is not yet known that how long we can still passage it, but ideally it should not be passaged for very very long time, so because it changes a lot of phenotypic because it causes a lot of phenotypic differences in those cells. But yes, but people have been using it, and they are like so it is not like how many cell lines after you have to stop it. Just we have to check the morphology of the cell. If it is fine, you can continue working with it until and unless it doesn't die. So yes, so the option so this question everybody has written the correct answer only. Okay, so yes, so that is, I think, it for today. um, so yeah, so Khushi is raising hand. Can you you want to ask your question? Khushi? Samiksha, about the previous sessions, I think only one session has been held, right? Till now, one session yesterday was held. So I think they will make it available. So I am not sure. I will make the slides and the recorded version of the lecture available by today or tomorrow. So yeah. Thank you, everyone. So yeah, so anybody else has any other question, or else we will just say bye for today and again meet next week. So I hope it was like worth your 2 hours of time. Okay, great. Samad is asking, ma'am, the HeLa cells are cancerous and irregular, so why after some years it will become primary because of not yeah, so there is lot of reasons. Cells get mutated, they are exposed to different kind. So there is handling like problems, like the way you will be handling it is very different from the way Western country people they are handling, or how even lab to lab it varies. I mean, the cell culture techniques are very lab dependent. So in our lab, whatever we are following, whatever methods we are following, it might not be same for all the labs in our department. So that way it is like very, I mean, difficult to say that when the cells are getting mutated or when the cells are deviating from their original type. So that's it. Kushi, next is again on Monday. So I will be taking classes every Monday from 6 p.m. to 8 p.m. Okay, so thank you everyone. Bye bye. Again, we'll meet next week. Okay, bye.